Okay, in summary, the paper established a robust strategy for driving human colonic organoids. And by applying this 3D differentiation strategy to FAP-induced pluripotent stem cells, they developed a disease-in-a-dish platform to monitor the overactivation of Wnt signaling associated with the hypoproliferation of colonic epithelial cells, highly relevant to colorectal cancer. They also found that a read-through drug, genedicin, could rescue APC protein expression levels, decrease Wnt overactivation, and block colonic epithelial cell hypoproliferation specifically in FAP colonic organoids without compromising the normal phenotype of wild-type colonic organoids. And in a mouse model, APC restoration promoted cellular differentiation and re-established crypt homeostasis in colorectal cancer, suggesting the translational potential of this organoid platform and its clinical application to, such drug candidates of precision therapy of individuals with colorectal cancer. There are some limitations with the study. While, at least in my opinion, the methodology and the procedures are very, very thorough, they can't account for some fundamental issues with the organoids. The use of these mini-guts lack peristatic contractions from smooth muscle of in vivo models, and the organoids are only as good as the surrounding matrix. However, the matrix used here is a lot less dynamic than what we would see in actual human clinical cases. The colon would also have access to blood and microorganisms because it is the colon after all. So as this is, it would prevent the effectiveness of this model toward investigations of toward investigating diseases involving infections or just anything with a more dynamic system. So a possible direction for the study would be to try and improve kind of the yield and the translational applicability, so to speak, of the organoids by maybe modulating some of the stiffness of the matrix during those 48 days of development. And this might change results as the cells are taking in more dynamic environmental signaling inputs. Okay, and with these results, precise methods can be used to derive these colonic organoids without the need for surgical resection of tissue. This would make it a lot easier to harvest and test out diseases of the colon, such as colon cancer, polyposis syndromes, colitis, or irritable bowel syndrome. And taken as a whole, this also sheds light on how these oncogenic cell types proliferate and provides one strategy to ameliorate that unnatural upregulation of Wnt signaling without affecting normal cell types, such as was the problem for XAV939 and rapamycin.